Hello friends, how are you? Hope all you are fine. In these lockdown conditions, I am discussing a new topic with all of you, that is plant tissue culture. This topic is very important and very interesting in point of view your examination. So we are going to discuss all the points, necessary points related to the plant tissue culture. So we are going to discuss the definition of plant tissue culture. What is meant by plant tissue culture? So lots of time in exam this definition was asked for 2 marks. So plant tissue culture is a technique of growing plant cells, tissues, organs or seed or whole plant in sterile environment in a nutrient media. So in very simple language, plant tissue culture is a technique in which the single cell can be converted into the tissue or organ or whole plant using sterile environment and using a nutrient media. So plant tissue culture is an important technology. People are preparing plant tissue culture. So why there is a need? Why there is a use? What is the use? What is the importance? What is the benefits of plant tissue culture? So why plant tissue culture is more important as compared with our traditional method? So we are going to study all the points one by one. The first is, using this plant tissue culture we are able to produce clones. These clones having a particular desirable characters. Maybe these characters related to the flowers, character related to the fruits. Means whatever the desirable characters which we want, we can include this character, and the character which we don't want, we can eliminate this character using this plant tissue culture. That is the most important characteristic feature of PTC. The next is, using this plant tissue culture, we can produce mature plant in a rapid manner, in a fast way. Means we can produce quick mature plant. So friends, in traditional method what we are doing? We are taking seed. These seeds we are sowing in a soil. After sowing, these seeds get water. Then it takes different type of nutrients from soil, and after getting all these favorable condition, this seed break its dormancy and there is a formation of plumule. This plumule grow and then after that we will get mature plant. So to complete all this process there is so many huge time is required. But in case of plant tissue culture, we can avoid this time period. We can minimize this time period. So in plant tissue culture, we can produce quickly, fastly, rapidly mature plant. The next point is, using this plant tissue culture without seed or with the absence of seed or with the absence of pollinators, we can able to multiply, we can able to produce the different type of plants. Means without seed or without pollinators we are able, means fertilization process, pollinating process is not essential in plant tissue culture. After that, next one is the regeneration of whole plant from the plant cell that have been genetically modified. Yes, genetically modification, I already told, we can produce clones. Means whatever the character is present in a parent plants, the same type of character is in a progeny. So that is called cloning, similar types characteristic plants or similar type of characters from parents to the progeny. So we can modify this regeneration. Means whatever the character which we want, we can regenerate this type of character. In picture C, This flower have purple color. Generally flower have white color whenever we are going to the market for purchasing this type of flower. So the flower have white in a color. They are available in a white color. But we can change its color. That is called regeneration in genetics. So plant tissue culture is also able to change the genetic structure of plant. After that, some other there which are going to study in the next slide. Using this plant tissue culture, we can also able to produce disease free progeny. So how using this plant tissue culture we are able to produce disease free progeny? So listen friends, plant tissue culture, these techniques we are generally doing in a sterile environment. Sterile environment means environment which is free from bacteria, which is free from dust, which is free from viruses, which is free from all living organisms, all living bacteria. So in that sterile environment we are doing these processes, means ultimately there is a less chances of transmission of disease because there is no bacteria, no viruses, no dust, no any foreign matter. So due to this type of condition, there is a less chances of transmission of disease. So whatever the progeny, whatever the plants we are getting, that is most of time disease free. Next one is what? Using these plant tissue cultures, we can also able to produce plant which have low chances of germination or low chances of growing. For example, orchid. If you take the orchid plant or orchid seed and these seeds sow into the soil, so it takes a huge time for the germination. Again, nephanthus, it means you are doing a work on 100 plants, from that only few plants are growing well. Means this kind of plants have low chances of germination and growing. But we can avoid this problem using plant tissue culture. Then next most important thing is what? In plant tissue, using this plant tissue culture, we are able to produce clean stock. Clean stock means stock which is free from of disease. This clean stock we can also further use in horticulture techniques and agriculture techniques. So these are the different uses of plant tissue culture, and that's why plant tissue culture having lots of importance nowadays. So before studying about plant tissue culture, there is a need to study some term, some definition, some terminology. So the first term, that is explant. So see in a picture some petri dishes, you can observe in that slide, and in that petri dishes some leaves. These leaves are not actually the complete leaf, that is the part of leaf, and that is called excise piece. So explant is nothing but an excise piece of differentiated tissue. Explant is nothing but a part of or piece of differentiated tissue or organ. So friends, what is meant by differentiated tissue? So this meaning of differentiated tissue we are going to learn in the next term. Next term is a callus. Again in petri dish you can observe irregular mass of cell. That indicates the callus. Callus is nothing but unorganized, undifferentiated mass of plant cell. Generally these callus cells or generally this unorganized mass is made up with parenchymatous cell. As I told you what is meant by differentiation, we are going to study on the next slide. So now we are going to study what is meant by differentiation. That was very important definition. There is a chances for asking this definition for 2 marks. Define differentiation or what is differentiation. Although if it is not asked in exam, but still This concept was very important. So we are going to talk about the differentiation. During plant cell development, cell becomes specialized for a particular function. Yes, every cell have their own function. This process is called plant cell differentiation. Every cell have their own particular function. That is the differentiation. Means xylem phloems are working for the transportation as a transportation tissue. Parenchyma is working as a photosynthetic tissue. Sclerenchyma is working as a supportive tissue. Okay, so every cell, every tissue have their own particular function. Means they have different function. The different cells have a different function. That is called differentiation. Means all the plant cells are not similar and they are not performing a similar type of function. Getting or not? See the picture. There is a picture of plant, and from that plant they are taking a one cell. That is called undifferentiated plant cell. This single cell, okay, this single cell is called as a undifferentiated cell. And we know what is meant by plant tissue culture. Plant tissue culture is a technique using a single cell. We can produce a whole plant using sterile environment and nutrient media. So this undifferentiated cell get converted itself into the different cell. That is called differentiation. So means the same cell is present in a leaf cell, this same cell present in a transport cell, this same cell present in a parenchymatous cell, this same cell present in a sclerenchyma or parenchymatous cell. That is called differentiation. Hope you will understand the meaning of differentiation. The next de-differentiation, that is exactly opposite to the differentiation. In differentiation, what we are studied, the same cell, the single cell are eligible to get differentiated into the different cell, and exactly vice versa, exactly opposite to the differentiation we have to talk in this definition. So de-differentiation is the phenomenon of matured cell reverting to meristematic state to produce callus. Callus cell or the cell which is present into the callus, this unorganized mass having capacity to go for the differentiation. So see in that picture, exactly opposite process they are showing. From leaf they are taken out a particular cell. This cell have their own particular function, means they are differentiated cell. Using the auxin and cytokinin, this differentiated cell get converted into the proliferated or callus formation cell, means that is the de-differentiation. Now these cells are a simple cell. Callus formation or callus formed cells are the simple cell. They don't have their specific function, means there is a de-differentiation. There, which are going to study in the next slide. Using this plant tissue culture, we can also able to produce disease free progeny. So how using this plant tissue culture we are able to produce disease free progeny? So listen friends, plant tissue culture, these techniques we are generally doing in a sterile environment. Sterile environment means environment which is free from bacteria, which is free from dust, which is free from viruses, which is free from all living organisms, all living bacteria. So in that sterile environment we are doing these processes, means ultimately there is a less chances of transmission of disease, because there is no bacteria, no viruses, no dust, no any foreign matter. So due to this type of condition, there is a less chances of transmission of disease. So whatever the progeny, whatever the plants we are getting, that is most of time disease free. How this totipotency work out? So adult plant cells are totipotent. They have the ability of totipotency. Okay. So this is possible to collect the cell from mature plant and use those cells to produce clone of that plant. So see in a figure, you can observe in that picture, we are taken out a root of carrot plant initially, and some cells we are isolated from that root of carrot plants, and these isolated cells we are transferring into the growth medium. This growth medium is also called as a nutrient medium. Growth medium, nutrient medium, these are the similar term, these are the synonym of each other. This growth medium or nutrient medium may be liquid, may be gel, may be semi-solid, may be solid. According to the need, according to the type of culture, we are choosing a particular type of growth medium. So this isolated cells which we isolated from the root of carrot plant is transferred into the nutrient media. So as we put up into the nutrient media, so this cell start to take the nutrients from that growth medium, and after taking all the necessary growth medium or after getting all the necessary nutrients medium, it started cell division. Okay, so cell divide and redivide, divide and redivide, and there is a formation of callus. Then this callus is get converted into the young plant, and then this young plant is get converted into the adult plant, and this adult plant having the similar type of characters of the initial plant. So that's why we are saying that it is a clone. Okay, that's why we are saying that the cells have ability to produce clone plant of their cells themselves. Okay, the next basic technique of plant tissue culture. So lots of time the big question was asked: describe plant tissue culture, give basic techniques of plant tissue culture, note on plant tissue culture, describe the method of plant tissue culture. Okay, so before studying method of plant tissue culture, we are just going to talk on some basic techniques, on some traditional techniques of plant tissue culture. So it was quite simple. So you just observe on a picture. First we have to take explant. Explant means part of plant. This explant may be bud or seed or root or leaf or any other part of plant. Then from that explant we have to isolate some single cells using trimming. Trimming is a technique. Using this we can separate out some cells of the explant. Now these cells are putted into the surface sterilization. After the surface sterilization, it was washed 3 times in a distilled water. Okay, and after washing, it is putted into the nutrient media. So you can able to produce a number of cultures. You have to take out just single cell. You have to put up this single cell into the nutrient media, and after doing this, you have to keep this cell plus nutrient media in an incubator for incubation purpose. Incubator is nothing but the laboratory instrument which have capacity to control humidity and temperature. Means on control humidity and control temperature we are going to incubate. The incubation period is also different. So according to the culture type, sometime it is for 12 hours, sometime it is for 24 hours, sometime it is for 48 hours. Okay, so for the like that we have to incubate this nutrient media in an incubator. So it is called incubation. So in incubation what happen? The cell take the nutrient media, utilize the nutrient media and start division. There is a cell division. Okay, so as you think the cell division is going in a proper way, there is a formation of callus culture, there is a formation of or growing of your culture, so you can also reculture it. It is called subculture. Okay, so that is the basic technique of plant tissue culture. So in modern techniques or in modern days or in a nowadays, the people performing all this plant tissue culture technique in a aseptic condition means whole room or the whole area is a sterile one. For that they are preparing the aseptic lab or aseptic chamber using HEPA filters. And nowadays people are also sterilizing eggs plant. In basic technique we studied that the eggs plant are not sterilized, but in the new technique the sterilization process starts from the eggs plant. So for the sterilization of eggs plant, usually they are using alcohol, sodium or calcium hypochlorite. Such a type of chemical are used. Generally, instead of alcohol, sodium or calcium hypochloride is more prominently used. Alcohol is also used, but there is a one disadvantage by using alcohol. If we are using alcohol for the sterilization of eggs plant, there is a shrinkage. If the flower or leaf we are using, so flower or leaf, it gets shrink in alcohol. But in case of seed or in case of stem, alcohol is not giving any type of disadvantage. There is a no shrinkage in case of seed or in case of bark or any hard material. Okay, so it is all about the plant tissue culture. The next point is what? If you are using a liquid media, so most of time that is the cell suspension culture, and if you are using a semi-solid or gel type of media, so that is most of time callus culture. So now we are moving towards the types of plant tissue culture. So there is a lots of types, so many types of plant tissue culture we are discussing one by one. But listen, generally if the liquid, if the nutrient media is a liquid, then we are going to prepare cell suspension type of culture, and if the nutrient media is a solid, not solid actually, the gel like or semi-solid, that time we are going to produce callus culture. Callus culture is a most important type because it is the intermediate step. Okay, for lots of culture first you have to go for callus culture, and using this callus culture you are going to again you are able to produce the particular type of culture. So how subculture works, how you can produce subculture? So see, as culture grows, pieces are typically sliced off. Means as the culture grows, you can put out a particular piece which is sliced off and transferred into the new media. To allow for the growth or alter the morphology of the culture is called subculture. The next one is very important type again, that is types of plant tissue culture. Actually we have to learn its meaning, its method, its example, all these. But before going to learn in a detail, we are just introducing this culture. We are just going to learn its definition. So first is callus culture. So all of you know that callus is nothing but the undifferentiated and unorganized mass. Okay, so what is mean by callus culture? The culture of undifferentiated tissue from egg plant which is de-differentiation in vitro to form callus. De-differentiates. Listen, plant tissue culture is a vitro technique. Plant tissue culture is a vitro technique. All the process happens outside the body. Then second culture is a organ culture. So as its name indicate, organ culture means culture of any organ. Means sometime we are using seed culture, seed, or sometime we are using ovary, ovule, anther. So those part we are using, that is this type of culture. Culture of isolated plant organ is referred as organ culture. So the organ is a embryo, that is embryo culture. Organ is a seed, it is a seed culture. Organ is a root, it is a root culture. Organ is a anther, it is a anther culture, ovary culture, ovule culture, endosperm culture. Okay, so like that is all about the organ culture. Next is organized organ culture. So means if we are taking the whole organ, whole organized organ, if we are using whole organized organ for the culture purpose, it is called organized organ culture. Then cell culture, the culture of isolated individual cell obtained from an explant or tissue or callus, regarded as a cell culture. These culture are carried out in a dispersion medium and are referred as a cell suspension culture. Protoplast culture. Protoplast culture is also one of the important type of culture. Before learning about the protoplast culture, first see what is the difference between cell and protoplast. Cell along with membrane, cell membrane is utilized, then it is called cell culture. And if we are removing its cell wall, cell membrane, okay, protoplast means what actually? The part which is remain after the removal of cell wall, that is called protoplast. If we are using such a type of material for the culture purpose, it is called protoplast culture. So that is all about the different types of plant tissue cultures. Besides that, some again there are some types, that is hairy root culture and hybridization culture or endosperm culture or endospermic hybridization, and all these remaining things are also we are going to study. But before that, we are going to study nutritional requirement of plant tissue culture. Yes, nutritional requirement is very important. Media used in plant tissue culture contain nutritional component. Nutritional components are essential for the growth and development of cultural tissue. Actually, the success of your plant tissue culture depends on your nutritional requirement or nutritional media which you are choosing. So though this process takes place in a vitro, means outside the body, so we have to provide all the necessary growth requirements or the growth material or the essential material or the food material or the all type of vitamins, hormones, we have to provide it. So the combination of all these is called nutritional media or the growth media. So what is the content, what is the composition of this nutritional media or growth media? We are going to study this. It contains some macro inorganic nutrients. It also contains some micro inorganic nutrients. It contains iron, vitamin, carbon sources, organic nitrogen, plant growth regulator, and along that agar. Okay. Now in next slides we are going to study all this nutrient media composition one by one. First, macronutrients. Macro means which is required in a somewhat large amount. Okay, so they categorize the nutrients in two types, macronutrients and micronutrients. Means nutrient which is required more than 3 ppm, part per million. ppm means part per million, which is required more than 30 ppm. They are coming under the category of macronutrients. And the nutrient which is required less than 30 ppm, they are coming in the category of micronutrients. So macronutrients are nothing but the much essential material, that is nitrogen, phosphorus, potassium, sulfur, magnesium, calcium. All these are the macronutrients. They are performing very important function of the cell. They are responsible for the protein synthesis. They are responsible for the cell wall synthesis. They are also responsible for the membrane integrity. They are also responsible for the enzyme and cofactor synthesis. So that's why macronutrients is very essential at the time of production of nutrient media. Then next, so how subculture works, how you can produce subculture. So see, as culture grows, pieces are typically sliced off. Means as the culture grows, you can put out a particular piece which is sliced. as I told you what is meant by differentiation, we are going to study on the next slide. So now we are going to study what is meant by differentiation. That was very important definition. There is a chances for asking this definition for 2 marks. Define differentiation, or what is differentiation. Although if it is not asked in exam, but still this concept was very important. Organic nutrients means vitamins. So we already know that vitamins are very important. For human beings also there is a vitamins are very important. If vitamins are in a deficiency state, we are going to suffer different type of disease. So plant also have ability, the cell also have ability to synthesize some vitamins. These vitamins are essential for so many biochemical reaction. These vitamins are essential for so many metabolic reaction. So culture cell also have capability to produce some vitamins in some extent, but they are not having the capacity to produce this vitamin in a sufficient level. So we have to supply this vitamin in exogenously. There is a need of exogenous supply of vitamins for the optimum growth at the time of plant tissue culture. So generally we have to provide these vitamins like thiamine, pyridosine, nicotinic acid, and some vitamin B complex. Okay, so these vitamins we have to provide in our nutrient media for the well growth, well development of our cells. Then hexitol. Most of tissue culture media have this component. Generally some contain mannitol, some contain sorbitol. Mannitol, sorbitols are also example of hexitol. They are essential for seed germination, sugar transport. They are also useful for the carbohydrate metabolism. They are also useful for the membrane structure. They are also useful for the cell wall formation. So that's why hexitol also have their important role in a plant growth and development. That's why hexitol is also one of the important component in growth media or in a nutrient media. Then amino acids. All of you know that amino acid is responsible for the protein synthesis, and protein is a primary food material for all living organisms. Carbohydrate, protein, lipid, these three are the primary metabolites, primary nutrient media. So they are called as a primary metabolites. So plant require proteins for their well growth. So this whole plant tissue culture process takes place in a vitro, means outside the body. So cells are not able to go for the photosynthesis, so they cannot produce their own food material. So we have to provide proteins, carbohydrate and lipids. So proteins we have to make them available in the form of amino acid, and after using these amino acids they are eligible to convert them into the protein by means of protein synthesis. So we have to provide this amino acid in the form of glycine. Then next is de-differentiation. That is exactly opposite to the differentiation. In differentiation what we are studied, the same cell, the single cell are eligible to get differentiated into the different cell, and exactly vice versa, exactly opposite to the differentiation we have to talk in this definition. So de-differentiation is the phenomenon. Then gelling agent, that is agar. Agar is also coming under the category of carbohydrate. It is obtained from the red algae from seaweed. That is called Gelidium amansi or Gelidium glycilia, belonging to the family Gelidiaceae. So why they are using this agar? Because there is a two reason. Agar is itself a carbohydrate containing material, so it can also be utilized as a carbohydrate containing food material. Along that, it has ability to convert this growth media or nutrient media into the gel. Agar have capacity as a gelling agent. So if you are taking 1 gram of agar into the water and you boil it on 100 degrees Celsius and then you cool it at the room temperature whole material get converted into the steep gel. That is the power, that is the function, that is the characteristic feature of agar. So that is all about the different types of plant tissue cultures. Beside that, some again there are some types, that is hairy root culture and hybridization culture or endosperm culture or endospermic hybridization, and all these remaining things are also we are going to study. But before that, we are going to study nutritional requirement of plant tissue culture. Yes, nutritional requirement is very important. Media used in plant tissue culture contain nutritional component. Nutritional components are essential for the growth and development of cultural tissue. Actually, the success of your plant tissue culture is depends on your nutritional requirement or nutritional media which you are. Then hexitol. Most of tissue culture media have this component. Generally, some contain mannitol, some contain sorbitol. Mannitol, sorbitols are also example of hexitol. They are essential for seed germination, sugar transport. They are also useful for the carbohydrate metabolism. They are also useful for the membrane structure. They are also useful for the cell wall formation. So that's why hexitol also have their important role in a plant growth and development. That's why hexitol is also one of the important component in growth media or in a nutrient media. So today we discuss that much point. Today we discuss what is meant by plant tissue culture, why it is important. We also discuss some definition, means what is meant by explant, what is meant by callus, what is meant by differentiation, dedifferentiation, redifferentiation, totiopotency. Then we also discuss different types of plant tissue culture. We also talk about basic techniques of plant tissue culture. And in detail, we studied composition of nutrient media or growth media. There is a high chances asking question on composition of nutrient media. Discuss the composition of nutrient media. Note on composition of nutrient media. Okay. So it was quite easy and you are easily able to write on that. The remaining part we are going to study in a part 2 of plant tissue culture. Hope you understand. After hearing, after learning, after reading up to that point, if you feel, if you have any doubt, you can contact me, you can message me on WhatsApp, okay? And stay home, be safe, okay? Now we will meet after some time in part 2 of plant tissue culture. Thank you.